Scientists uncover 18-million-year-old proteins in fossil teeth. Imagine uncovering secrets from 18 million years ago, secrets hidden not in bones or fossils, but in the tiny remnants of proteins trapped inside ancient teeth. A groundbreaking discovery by scientists has revealed that fossilized teeth can preserve proteins for millions of years, rewriting the rules of molecular preservation and opening a new window into the lives of extinct species. Proteins, the building blocks of life, typically degrade over time, making them nearly impossible to trace in fossils. However, a team of researchers from Harvard University and the Smithsonian Museum Conservation Institute made a startling find: intact protein fragments preserved in the enamel of 18-million-year-old mammal teeth unearthed in Kenya's Rift Valley. This breakthrough challenges the long-held belief that mature tooth enamel, the hardest substance in the body, contains virtually no proteins. Teeth are remarkably durable, akin to tiny rocks in our mouths, explains Daniel Green, lead author of the study published in Nature. Their geochemical record can reveal an animal's diet, water sources, and environment, essentially a blueprint of its life. Using advanced proteomics techniques like liquid chromatography tandem mass spectrometry, LC-MS/MS, the team detected a diverse array of proteins in modern teeth, then turned to fossils. But here's the surprising twist. Ancient teeth from early elephants and rhinos, with their thick enamel, up to 3 millimeters thick, yielded peptide fragments, short amino acid chains, that survived for 18 million years. Nobody has ever found peptide fragments this old before, notes Green. It's kind of shocking. Prior records topped out at 3.5 million years, making this discovery a five-fold leap backward in time. So, how did these proteins defy decay? The key lies in enamel structure, which acts as a protective vault. We're not just finding one or two proteins, we're seeing dozens, Green emphasizes. These proteins aren't random survivors. They represent a proteome linked to biological functions, offering clues to the animal's physiology, diets, and even evolutionary relationships. This isn't just about ancient elephants, says Emmanuel Ndiema of the National Museum of Kenya. It's about unlocking molecular traits of extinct species, from dietary adaptations to diseases. We can now place them on family trees using proteins, much like DNA. For example, proteins could reveal if an extinct rhino species was more closely related to modern horses or cows, a question previously unanswerable with bones alone. And this is where the discovery gets controversial. If proteins can survive this long, does it challenge our understanding of how life's molecules degrade? This raises big questions about preservation mechanisms, notes Kevin Uno, a co-author. Why did these proteins last so long? The answer could upend theories in biochemistry. Skeptics might argue that contamination or lab errors explain the findings, but the team rigorously ruled out such possibilities. The implications are vast. Paleobiologists can now reconstruct evolutionary histories with molecular precision, even for species with no living relatives. As Green puts it, you could theoretically take a tooth from any extinct animal and rebuild its place in the tree of life. This project began as a small experiment, but grew into a landmark study funded by the National Science Foundation and Smithsonian's Conservation Institute. As Uno reflects, we were blown away by how much was preserved. It's a whole new frontier. Reference: Green et al., 18 million years of diverse enamel proteomes from the East African Rift, Nature, DUI, 10.1038/S41586-025-09040-9. Your turn asterisk. Could protein preservation in teeth revolutionize paleontology? Do you think these findings will spark debate about the limits of molecular fossilization? Share your thoughts. 24-million-year-old rhino tooth discovery, a game-changer in paleoproteomics. Imagine uncovering a 24-million-year-old rhino tooth. That's exactly what a team of scientists from the University of Copenhagen has done. This discovery, made in the frozen wilds of the Canadian Arctic, is a game-changer for biological research. The team, led by researchers Ryan Patterson and Enrico Cappellini, found ancient proteins in the tooth, which are 10 times older than the oldest DNA ever sequenced. The secret to this remarkable preservation? The toughness of tooth enamel, which acts like a molecular time capsule, holding onto proteins for millions of years. The rhino tooth was extracted from Nunavut's icy Houghton Formation. This find will help scientists determine where this particular breed of rhino fits in the family tree. Initial speculations suggest it might be somewhere between a woolly rhino and the Siberian unicorn. Yes, you read that right, there was a creature called a unicorn, but it was far from the magical, Lisa Frank-looking white horse we imagine. It was actually a hairy rhino with a gigantic horn. But here's where it gets even more fascinating. In Kenya's Turkana Basin, another team led by scientists from the Smithsonian and Harvard discovered ancient proteins in fossils up to 18 million years old. What makes this discovery particularly noteworthy is that these molecules were preserved in the blazing heat of a tropical desert, challenging the assumption that proteins can only survive in deep freeze conditions. Consider this your introduction to a whole new field of research called paleoproteomics, defined in a 2022 research paper as the study of ancient proteins. This field is set to fill in the gigantic chronological blind spots left by DNA, which tends to degrade after 1 million years. Proteins, on the other hand, are tougher and more persistent, capable of telling the story of the creature they lived in even after all these millennia. And this is the part most people miss, the potential for paleoproteomics to revolutionize our understanding of ancient life. But here's a controversial question. Could this new field eventually replace traditional DNA analysis in certain contexts? What do you think? Share your thoughts in the comments below.